So I think it's visible.
Yes, sir, it's visible.
Yeah. So welcome the participants for this last session of today's GIAN program, where we will be giving you a detailed idea about the uranium biokinetics in human adults and protracted ingestions of groundwater. So, like in the previous lecture, I have discussed various techniques for the estimation of natural radionuclides in air, water, and soil. This particular lecture will be dealing with the uranium biokinetics, which we are using. We are one of the pioneer lab in this region, or especially we could say in India, who are using the biokinetic modeling for the estimation of different doses to our different organs due to the concentration of uranium in groundwaters. Now, if you see the outline of the talk, we will be discussing a little bit about the routes of exposure, uranium in groundwater, and then biokinetic modeling, and some other different models of ICRP hair compartment model, and finally one case study I will be presenting for the estimation of uranium concentration in water, and finally calculations of the doses. Now again a similar type of a picture. Once we say it is a radiations are continuous from the lower energies to a very higher energies, starting from radio waves, UV, visible, infrared, microwaves, extra. So, and generally they are divided into two broader categories. One are the ionizing, another are the non-ionizing radiations. And out of these ionizing as well as the non-ionizing radiations, generally the ionizing radiations have more impact. Like the mobile phones which we are using, they are giving us the non-ionizing radiations. So though we are having mobile phones continuously with us 24 hours rather, I could say nowadays, so they are giving you some impact, but their impact is not so high as compared to the exposure from the ionizing radiations. Because the ionizing radiations give you more impact as compared to the non-ionizing radiations, radiation exposure, which I was discussing in my previous lecture also, that is as far as the United Nations report is concerned, UNSCARE's report is concerned, that is 2.4 millisievert. And out of this, and out of this 2.4 millisievert, this nearly 1.15 millisievert or even up to 1.5 millisievert is only due to the concentration of radon. So there are different routes of exposure to our body, that we could have some external exposures, we could have some internal exposures. So external exposures, if you see from the figure, it is giving you an idea about how the exposure is entering, like you have inhaled something, it is entering to your body through your inhalation process, then it is depositing within your lungs and even your kidneys, even your intestines, and finally affecting you. There are some internal exposures also. So internal exposure means inhaling radioactive material in air, ingesting contaminated foods and drinks, then inadvertent ingestions, absorptions through the skin or open wound. So that is also some of the types of internal exposures are there. Now, if you see the quantified intake by ingestions or inhalations, so based upon your quantified intake, that how much you have taken as input, either you have ingested something or you have inhaled something, we use the different models which we call as a biokinetic as well as the dosimetric models to have an estimation of the quantified intake of ingestions or inhalations. And then we use these models to estimate the mean absorbed doses in the organs and tissues. So like if I drink 2 liters of water per day, so my exposure will be different from a person who takes 4 liter of water per day for a same type of a body weight, for a same type of a exposure. The reason is because the natural uranium contamination in 2 liters of water and 4 liters of water is different. So depending upon the type of intake, that is the quantity of intake, some of the factors are being applied. Then if you apply radiation weighing factors, then from the absorbed doses, you could calculate the equivalent doses to an organs or tissues. So that means my lungs, my heart, my chest will be behaving differently for a same type of intake. And then applying the tissue weighing factors of my different tissues, I could finally calculate the effective doses to the different organs. So which we generally call as a gonadal doses. So like if I drink 2 liters of water, my exposure to lungs will be different. For a same intake of 2 liters of water, my exposure to kidneys will be different. For same intake of 2 liters of water, my exposure to bones is different. So depending upon the different weighing factor, radiation weighing factors and the tissue weighing factors, my exposures are being calculated in details. And you can't say that if the rate of concentration of uranium in water or 60 ppb or 60 microgram per liter, it will be giving a same impact on my lungs, it will be giving a same impact on my heart. No. So for different organs, the same concentration will give a different type of impact. For different tissues, the impact will be different. For different age groups, the impact will be different. For different sex, the impact will be different. So for different concentration of different salts or the exposure in the water, the effects will be different. So there are two parameters. One is if the concentration of uranium is different in different soils or in different waters, impact will be different. It is very true. But for a same concentration of water, same concentration of uranium in water, the impact on different organs will be different. So it is very important to study the impacts of different organs instead of saying that this much concentration will give you this much of impact, because it is a general estimate that if the exposure is this much, my impact will be different, my impact will be this much, my effective doses will be different. But for my heart, the doses will be different, my lungs, the doses will be different, for my bones, the doses will be different. So it is very important to calculate the different tissues weighing factors also while measurements are being carried. Now if you see the uranium, it is present since the Mother Earth and half-life is, the life of Earth. And from the very beginning of Earth, uranium was present, and still the age of Earth is the half-life of uranium. That is for 4,500 million years. So uranium is groundwater. Why it is important? So it is a lithophilic element which is having five different rather valence states: 2 plus, 3 plus, 4 plus, 6 plus. And out of these 2, 3, 4, 5, 6, the 4 is insoluble in water and the hexavalent is soluble in water. So if you are having more hexavalent contribution valence states present in that particular water samples, so that is once, even in the mountains, even in the glaciers from where the rivers or the channels are flowing, so if the mountains are having more 6 plus valence state, so it will be soluble in the water running through across that mountain, across that channel, across that glacier. So source may be in the Himalayas, but impact may be in the Gangas. So source may be in the Himalayas, impact may be in the Calcuttas, where the deltas are there. So you can't say that if the concentration is high at a particular place, the source is also at that place. So source may be from different places. So that is why it is very important to have an idea of the geology of that particular area before reaching to some of the final conclusions. Because what happens that sometimes we say that this is the concentration and this is the impact, so this area is not good for living. So it is not there. You have to understand the basic idea that how the geology of that particular area is responsible, whether that geology is contributing or whether some different sources there which is contributing to the high concentration. And to your surprise, in my area, especially in Punjab, where there are a lot of cases of a high level of uranium concentration in Malwa region of Punjab, lot of studies have been carried out. Different researchers have published different reports and published different papers where they have said that in some areas of Firozpur, Faridkot, Mansa, Bathinda, the level of concentration of uranium is very high. But to your surprise, even within a same village, the concentration is high in some of the areas, and within the vicinity of 100 meters, the concentration is within the safe limits. So how it is happening? So it was very surprising for we people also that if the concentration of groundwater is high in a particular hand pump or a tubewell where we have extracted the groundwater from a particular place, within the vicinity of 100 meters, the concentration is very low. So then we have to take the help of the geologists, where we could make out some of the conclusions, but still it is not the perfect conclusion, I would say. But still, a lot of detailed investigations are required. That some of the areas in that particular regions, they were having some perched layers. It was a waterlogged area. So where water has been interlocked within some of the perched layers, and that perched layers, the water concentration was due to the high level of granites which were present in the geology of that area, and it has high concentration of 6 plus valence state which is water soluble, and with the mixing of granite with the groundwater, the concentrations were high in some of the areas. So that is why it is very important to understand what is the geology of that area, and then enters groundwater aquifer due to the natural geochemical processes like mineral dissolutions and the desorptions of absorbed uranium from the mineral surfaces. That is also important. Anthropogenic contributions, we have to understood that what are the anthropogenic concentrations in those areas, what are the industries, what type of industries are present in those areas, what type of activities are going on in those areas, whether they having some fertilizer plants, whether they are having some thermal power stations, whether they are having some nuclear facilities. So those things are also very important to understand to have an idea of the anthropogenic contributions. Then hexavalent species UO2 plus, so that is also important to understood. And then finally you have to understood that what type of contacts you are using, like whether you are drinking the groundwater or whether you are ingesting some food samples or whether you are coming into the dermal contacts. So that is important. Now, this principal sites of uranium depositions in the body, they are important organs like kidney, liver, and bones. So those are the three important parameters. And now the hairs, that is your hair present in your body, they are also the major contributor. So how it is dangerous, uranium is dangerous. So from the figure you could have an idea that you could have a high blood pressure, you could have some kidney diseases, you could have some reproductive functions, you could bear some bone cancers, you could have some kidney functions, you could have some lung cancers with the high concentration of uranium. Some are the chemical effects on the left side, radiation effects on the right hand side, which we have mentioned. And the sources of uranium in the groundwater, one is the natural sources of uranium in the groundwater, means like I said that you are having some geology of such mineralizations or rocks that your groundwater is naturally radioactive, like if you are having more granite in those areas, so that may be one of the reasons. Then there are some anthropogenic activities also. Anthropogenic activities means if you are using more pesticides, more agriculture fertilizers, again some waste of nuclear power plants or wars are there, then mining activities are going on, means if you are having some nearby mines areas, so those are anthropogenic activities, they may also contribute. And there are some cosmogenic activities also which are there but they contribute very less. So most of the contribution is due to the geogenic, that is weathering by steam streams of uranium rich rocks, which I have already told you that if your mountains are having more hexavalent components, so they are water soluble, so any water body which is traveling through that source, they will have more concentrations. Then there are some anthropogenic activities. Now, these are the source pathway receptor analysis of uranium. Like if you start from the very beginning, from the atmosphere, you could have concentration into the soils, concentration into the fresh waters, concentration into the urine and marine waters. And then from the soil and fresh waters, they are again interlinked with each other. You could have a concentrations to the groundwaters, you could have concentrations to the sediments, and then again from the third component, that is the marine waters, you could have concentrations to the sediments. So these are the spatial distributions which have been represented in the dotted block lines at the upper, which are the sources. So from this, it could be bioaccumulations or exclusions, and then you could inhale it from the sediments, and you could ingest it from the soil sediments and drinking water. And then from the bioaccumulations means if the plants absorb those high concentrations, or if the animal take the intake from those high concentration, say cattle feed, so they could again be given back to the human beings in terms of once we take those plants as our vegetables, and once we take milk products from those animals. So it could be ingestions of plants and the animal products, which could again give us the exposure. So we are having different environment, but from all the environment, the assessment or impact on humans is there. So we could see from this figure that from the spatial redistribution to the bioaccumulations, final concentration is coming to the receptor as the humans. So then there were different models which have been suggested by different scientists. The first biokinetic model was proposed by Bernard and Strunes in 1957. So basically they were dealing with the blood, bone, kidney and urine. Means how this uranium from the blood is excreted in terms of the excretion, either is a urination or a feces, and then how this concentration is distributed across different bones and other organs. So this was the initial model which was started. Like if you take an intake of water or an intake of high concentration of uranium, so how it will be excreted out. So the best way of excretion was the feces as well as the urination, which was understood at that time. So kidney was also considered the most susceptible organs to the radiation damage rather than the bones. So generally we say that if our concentration of uranium is high in terms of water concentration, so our kidneys will be mostly affected as compared to the other organs like bones. But then with the advent of new sciences and the technologies, the ICRP published new models which we call as ICRP2 biokinetic model of 1959 and ICRP systematic biokinetic model of 1995. So if you see from the figures on the upper model, from the blood plasma, the concentration was given to the bones as well as the kidney, and finally it could be excreted out in terms of urination or feces. But in the systematic biokinetic model, which was a revised model in 1995, which was published by the ICRP in the 69th publications, these all models are available free of cost on the ICRP website, International Commission of Radiological Protection's website. You could have or download those models or parameters from their publications, and finally you could understand what are the different advancements in different models due to the work done, of course, for a prolonged period of time, how they have interpreted that, how the intake of uranium concentration is affecting our different organs and how we are excreting it. Now, from the ICRP 1995 model, if you see, the concentrations have been distributed to different bones, and kidneys component have been also distributed, because sometimes we have a very high exposure immediately after we drink something, and later on our plasma go on excreting it, and finally with the help of urination and feces we are excreting out from our body. Then final important model with the advent of new sciences and understanding of technology was developed by W.B. Lee, which we call as a 2010 hair compartment model of uranium. So if you see from this figure, from the blood plasma, it is going to our livers, it is going to our small intestines, it is going to our upper large intestine, it is going to our kidneys. It's a two-way. You see the directions of the arrows. It is flowing two-way, means from the kidney again it is coming to the plasma, from the plasma it is going to the kidneys, from the liver it is going to the plasma again, from the liver it is plasma, it is going to the livers. So if you see the ingestion component, once we ingest something, means if we eat something, so it enters to our stomach, then from our small intestine it goes to the blood plasma, and again from the blood plasma it goes to our large intestine. So from small intestine it is directly going to our large intestine, and even from the blood plasma it is going to our large intestine, and then to the lower large intestine, and finally we excrete in terms of feces. That is, we have ingested something and we have excreted something. Now in terms of body or bones, which we call as a skeleton or a trabecular surfaces, cortical surfaces, so cortical volume and trabecular volume is very important, so bone density is very important. So again it is going to our blood plasma and finally going to the kidneys, and from the kidneys to the urinary paths, and from urinary paths to the urinary bladders, and finally we excrete in terms of urination. So these were the few models which were available at that time, that we could excrete either in terms of feces or we could excrete either in terms of urination. But the third component which was added at that time was the hair, that is our hair present in this body. So from the hair it was informed that, or from the model it was predicted that nearly 31.5% contribution of excretion is through the hairs. So which was very surprising. And it was also proved in some of the documentaries or in some of the researchers that if you are working in some high radioactive areas, so your hairs have a high concentration of uranium contaminations or those radioactive contaminations. So even these type of activities were used by some of the investigating agencies for collecting the hair samples from the barber shop of some of the scientists and estimating whether they are working in some restricted areas, in some nuclear areas or some radioactive exposure areas. So from the other soft tissues or from the hair, the excretion is nearly 30%. So that is why it supersedes all the ICRP's previous biokinetic models. And we now have three channels of excretion. One is the urination, another is the feces, and the third one is the hair. So this is one of the case study which I will be presenting to you, which we have done a survey of the biokinetic studies of uranium ingestions through groundwater in Chittorgarh district of Rajasthan area. So Rajasthan is in the western side of India, and most of the district we have tried to cover it. We have made a grid map of around 10 kilometer by 10 kilometer scale, and then we tried to cover the maximum grids depending upon the availability of the transportations in those areas. So we tried to cover some of the grids, and these are the few limits which you should understand. What are the few maximum contamination levels given by different agencies for the human beings. The WHO and the US EPA, that is the United States Environmental Protection Agency, has given a safe limit of 30 microgram per liter. So if your concentration is within 30 microgram per liter, it is safe for drinking purposes. So it will not be affecting you. So Anscare in 2000, they have given a limit of 9 microgram per liter. So microgram per liter is basically the concentration in ppb, that is parts per billion. And in India, the EERB has given a limit of around 60 microgram per liter. And now why there are ICRP has given a 1.9 microgram per liter as a safe limit. So of course it is working with the radiation workers, so they have been exposed for other radiations also. That is why the limit is very less. So from the water, if you get this much of exposure, because you are working in some radiation areas, so you are being exposed. So that is why the safe limits for the radiation worker is very less in case of ICRP, that is 1.9 microgram per liter. But for Indian people, generally it is of the order of 60 microgram per liter, and it has been calculated on the basis of criteria that how much water we generally take. Generally, for a case of adults, we take average intake of 2 liters. So based upon that 2 liters of intake, it has been calculated as 60 microgram per liter is a safe limit. Also, it depends upon some of the physical conditions or biological conditions of different ethnic communities. Like we as Asians or Indians have a little bit more safer limits as compared to the European countries. So that is why our limits are little bit on a higher side also. And this is the methodology which I will be discussing. That how we are going to measure it, and we will be giving you the live demo of this measurement also during the 5th day, where we will be using the different fluorines and calibration modes. Finally, we will be telling you that how you have been measuring these uranium concentration at a different parameters. So this is our sample holder, this exposure which cursor I'm showing. So this is our sample holder. So where we place our sample in a quartz container, and finally with this working principle of fluorescence of uranium complexes in aqueous medium, we measure the uranium concentration, and measurement time is very small. It takes around 1 second for an average of 256 pulses taken 5 times. And this instrument has a analytical range of measurement, that is 0.5 microgram per liter or ppb to 1000 microgram per liter or ppb. And with the help of this, we measured at different locations in our samples, and we found that apart from this one red colored sample, in most of the cases, the concentration is within the safe limits prescribed by the Atomic Energy Regulatory Board of India. But if you talk in terms of WHO also, there are 2 samples which are having higher values of uranium concentration. And if you take in totality, there are 3 samples only in 3 villages where the uranium concentration was higher. So what we tried to correlate it with the geology of that area also, which I will be discussing in our next lectures, that how the different concentration of soil samples is impacting the concentration of uranium contamination in water. So then we calculated the detailed analysis with the help of some geologists, and we studied in some of the areas. So we found that there are some geological parameters which are responsible for giving a high concentration in those areas, so which we'll be discussing in our next lectures. Now based upon this, We applied the hair compartment models to our concentration, and if you see from the hair compartment model, so we calculated the different organs component. So we calculated it for kidneys, we calculated it for liver, we calculated it for lungs, we calculated it for pancreas. So this is the concentration distributions which has been given or shown, that how it is being there. So if you see the values from this, the bone surfaces has around 38% of concentration, and then from the bone surfaces you could imagine that maximum retention is on the bones. So from this we concluded that out of the 32 water samples, the uranium concentration in 3 samples were exceeded the recommended action level or recommended well limits of WHO and US EPA, and only 1 sample exceeded the limits of Atomic Energy Regulatory Board. So, like I have told you that we have done some geological calculations also. So there are some biotite materials present at a depth of around 300 feet. So due to those biotite materials, the concentration was high in those nearby villages. So which is again a part of concern for us. So which act as a subsorption elements for the uranium. So those biotite materials were present in those areas. So we are we have extended our research in that particular area also, where we are working on some of the natural development of natural polymers, so which could remove those uranium concentrations from the groundwater samples, and they could absorb. And we have attained a very successful result with the help of some banana leaves, that we have developed some polymers with the help of banana leaves. We have extracted the cellulose from the banana leaves and then used that cellulose for the removal of uranium contamination in the water samples, and we have achieved nearly more than 90% of removal. So again, we will be discussing those things in our next lectures, when we will be discussing some of those materials. So we found that infants between the ages of 7 to 12 months are the most vulnerable to the radiation exposures among all the populations and the age groups. So the reason for this is like most of the infants at the ages of 7 to 12 months were depending upon the lactic mothers, and for lactic mothers the concentration or the water intake by their mothers is also impacting them. So they are being breastfeeded by their mothers, and finally they are being affected. So however, even for the most vulnerable segment of population, that is children, the toxicity hazards analysis provides reassurance that the predicted levels of uranium exposures is modest, indicating negligible chemical risk. So external hazard quotient for all the areas was less than 1. So that is, the drinking water is safe for the local populations. It's not so much dangerous. So some of the remedial studies of a urinal from the aqueous solutions using the cellulose extracted from the leaf of banana, which I was discussing. So I will be giving you an idea about that how we have extracted it. So we have collected some of the banana leaves. Then those were fresh collected, washed banana leaves were there, then oven dried so that the moisture content could come to the level less than 5%. Then we have crushed it. Final bioabsorbed cellulose from the banana leaves have been extracted, and final product is generated. So this is a little bit time consuming techniques, but I am just giving you an idea so that you could have an idea that how this banana leaves are useful for the extraction of uranium concentration. And then we have done different transmittance analysis, and we have done their XRD analysis. We have done some SEM studies to prove that the cellulose is being generated. And from the different batches, we have prepared the standard solutions of different uranium concentrations. For 10 ppb, we are having standard solutions. For 100 ppb standard solutions, we do the batch analysis for because once we do the batch analysis, we have to take a standard also. So we compare the water samples with our standard solutions, which we prepare as our buffers, with the known value of concentration of uranium. So for 10 ppb we have done, for 100 ppb we have done with those standard solutions, and we have measured the uranium concentration. And what we have found that the absorption, once this cellulose was put into the water samples, the absorption efficiency or efficacy was around 90%, starting from 60 to 90%, and it was not affecting with the absorbent dose also. And finally, contact time we have taken from 15 minutes to 60 minutes. It was giving us a fair results that no deabsorptions was occurring even after 60 minutes. So once it has absorbed within the 15 minutes, we keep it there for further 60 minutes and to see whether some deabsorption is occurring. But our results were very satisfactory, and no deabsorptions occurred even after 1 hour. So then from this study, we concluded that the greatest uranium absorption effectiveness in the aqueous solution was, the maximum which we achieved was 96.69%, and generally it's of the order of 90%. So absorption studies from the uranous ions from the aqueous solutions was influenced by various factors, pH, that is the percentage of hydrogen, ion concentration, and the applied doses. Of course, they are the responsible parameters, and we have kept the ideal pH value for this is 4.5. So the ideal parameter for achieving the highest absorption efficiency were at pH 4.5, because we have tried at different pH values. So at 55 ppb, strong ion concentration and 10 milligram applied doses of uranium solutions in 10 ml of water. So at pH 7, the lowest absorption efficiency was measured at around 16.65%, with 55 ppb of the starting ion solution and 10 milligram of applied doses. So the best value which we achieved was for the pH 4.5. The experimental data plot shows that no influence of contact times, which I have discussed, that no deabsorption occur even after 60 minutes of exposure. Then the cellulose which was derived from the banana leaves was found to be effectively removing the uranium ions from the aqueous solutions. So banana leaves can act as natural polymers. So we are working along with our colleagues in our institute by development of some other natural polymers with the help of some different plants and different naturally available plants, where we are extracting the cellulose from those plants and finally developing some of the bioabsorbents, so which are capable of removal of not only the uranium concentration but other heavy metals also. So we are working on cadmium, we are working on chromium also. So we are working on different heavy metals, where development of the natural polymers is giving us a good results, and we have published our results in very good papers. And of course, you are again welcome to visit our lab, and especially the chemical engineers and the chemistry people who are working in the development of the bioabsorbents. So we could collaborate with them so that we could have an idea that how we could develop some pallets type of polymers, so that we could fit those pallet type of polymers into the filters, and finally with the help of those filters we could remove the heavy metal as well as the uranium concentration from the water samples. And secondly, another thing which is important is like, of course, these are the facilities which are available in my lab. So we are having ICP-MS, we are having gamma ray spectrometer, AAS, LED fluorimeter, and RAD7. We invite the different chemical engineers as well as the chemistry people to give us more information about the development of the polymers, because though we are working in these particular areas of a natural environmental radioactivity, but important parameter is like how to remove it. So we could estimate it as a physicist, we could say that this is the concentration, but how to remove it, there the role of some chemical engineers and chemistry people is very important, because they are having good expertise in development of the polymers. So we have achieved some of the removal components or removal pallets we have developed with the help of cellulose nitrate or cellulose materials extracted from the banana leaves. We are working in some other natural polymers also, but still the lot of intimacy from the different interdepartmental activities is required to reach to the final conclusions. So that is also the basic purpose of research. You can't restrict yourselves to a particular area, because I cannot be expertise in all the fields. So if I'm expertise in measurement, so one should be an expertise in the removal. So it is not only one thing is important, that I could say that this concentration is very high in this area and this area is very dangerous. So I have to give some solutions also. So for those solutions, we have to collaborate. So of course, these are the atomic absorption spectrometers which is available with us, and we are measuring chromium, cadmium, lead, copper, manganese, zinc, and another even mercury and arsenic. So nearly 16 heavy type of heavy metals we are measuring, and we are giving the consultancy to the different food industry, chemical industry, even our local municipal corporations to have an idea about the concentration of heavy metals in the water. So of course, this is again a Agilent's plasma coupled spectrometer which is available in my lab. So you could always come and visit to us. So with this, we conclude for this lecture, and if any queries are there, so you could always ask, and we are free to answer those queries. And I am thankful to all the listeners, and I hope that during the next 4 days also, because this is a 5 days course, so next 4 days we will have your active participation, and I would request you that at least if you have some queries, you write down those queries in the chat box or the message box, so that we could note down those queries, and finally during the final day we would like to answer those queries, because it's a two-way process. So Government of India has taken a very good initiative where they have invited some of the experts from the foreign universities, so who are available to you, and they will be presenting their research. And but if you don't ask questions, if you don't have queries, so then by just one way of communication or a delivery is not important enough. So I would request all of you to be an active listener or active participant. And I once again thank all of you for being a patient listener. So with this, I would request Abhishek to conclude this first day of GIAN course. Abhishek, are you there?
Yes, sir. Thank you very much, sir, for sharing such valuable insights and making our session so enriching. I also thank all the participants for their patience, active engagement, and enthusiasm throughout the lectures and other sessions. We look forward to seeing you tomorrow for the next sessions with the Professor Shinji Tokonami, which will begin promptly at 10 a.m. So with this, I conclude.
Tomorrow's session is at 10 a.m., not at
Yes, sir.
10:30. Na? So
Yes, sir. So
in the
tomorrow.
morning we will be starting at 10 a.m. So I request all of you to join at least 5 minutes before or 10 minutes before the 10 a.m. So thank you once again, and hope for the best. Enjoy your winters. Thank you.
Okay. Thank you very much, sir. So with this, we conclude the session for the day one. So
Thank you. God bless you.
thank you, sir.